Good morning. Thank you very much for coming. I'm Fiona Crawford. I have the honour and privilege of being the President and CEO of the Roskamp Institute. And we want to welcome you here today. We do this event every year in honour of veterans and military, but we welcome everybody in the community to come and hear about our research. And in fact, we do have tours here every month on a Friday.
First Friday of every month.
I didn't even see Jane. You flew in specially. Hey. So I am just going to give a brief, hopefully very brief introduction to the Institute for those of you not familiar with us here, and then we're going to get into our program. I think you should all have the agenda. So the Roskamp Institute, we have been here since 2003 at this location here in Sarasota. We are a not-for-profit biomedical research institute. Started by myself, my colleague Dr. Michael Mullen, and Bob and Diane Roskamp back in the late 1990s up in Tampa, and then we relocated here in 2003, and we now have more than 60 scientists, clinicians, PhD students. We have a PhD program here, a very unique program from which we have graduated 8 students at this time, and we have 7 more in the program. And many of our students and young scientists will be your tour guides and the folks you're going to meet in the labs today if you go on the tour of the Institute. We target neurological and neuropsychiatric disorders, and our history is in Alzheimer's disease. I'm going to talk a little bit about that today. That's not going to be the focus of today's presentations. But our history is in Alzheimer's disease. Dr. Mullen and I were part of the team back in London in the 1990s that identified the first genetic causes of Alzheimer's disease. And so from there, our work progressed to identifying ways to tackle the disease and actually moving all the way forward into clinical trials. And you're going to hear about that today as well. So the Roskamp Institute funding, folks usually like to know how we get our funding. So I thought I'd put this slide up just to let you see there's a huge grant effort from the scientists in the Institute. A lot of our funding comes from the Department of Defense, from the Veterans Administration, from the National Institute of Health. We have, of course, our clinical trials and our neurology practice that you're going to hear from Dr. Keegan about today. And then we have the ongoing support of the Roskamp Foundation, for which we are extremely grateful. That's like a stable platform that everything else can build from. And then community support, which, thanks to Jane Thompson in the audience here, is an increasing piece of this pie chart for us. It's very important for us, our funding, of course. That sounds like such an obvious thing to say. But the work we do is not cheap. And I can assure you that every dollar that comes in the door here goes to the science. It all goes past me. And if I see a dollar coming in that isn't heading into the lab, I want to know why. So we are engaged in a number of different activities here. And you're going to see these today whenever you tour the Institute. We do behavioral analyses of our animal models. We look at the brain biochemistry, the blood biochemistry. We do neuropathological analyses. We can get down into amazing level of detail. We have all of these wonderful tools now. The mass spectrometry tools that we have enable us to look at the molecular level and get in and look at individual proteins and lipids. So we can take the blood from an Alzheimer patient in our clinic and compare it with somebody without Alzheimer's disease and look and see what's different in the lipids and in the proteins that are present in the blood of the person with Alzheimer's and the person who doesn't have Alzheimer's disease. These are all ways that we're working to find biomarkers for the conditions we're working on and to identify the molecules that are changing in response to the disease or in response to the injury that folks have sustained, and thereby identify targets with which we can intervene in those disease processes. The mouse model is kind of at the root of a lot of our work here, and I always like to say to folks when they come to visit the Institute, if we could do everything that we do here just with cells in a petri dish, we would do that. But it is impossible at this time. We do not have the skill, we do not have the know-how to model the complexity of the human brain in a petri dish. And therefore, for the work we're engaged in, we actually have to go to living animals. We have to go to a living brain because we cannot model the complexity, all of the different cell types, all of the ways the neuronal connections interact, all of the blood vessel input. We can't do that in cells in a petri dish in the lab right now. So we use mice, and we have mouse models, and our general approach is to develop good laboratory models, good mouse models for the conditions we're working on. Because then in mice, we can do something we cannot do, of course, in humans. We can actually look right after a mouse has been exposed to some sort of traumatic stress. We can look and see immediately what's happening in the brain. And then we can look again 3 months later, and again 6 months later. And in our studies, one of the things that we have done, and actually our team here is recognized because we have pioneered a lot of these very chronic studies in our animal models, because we're interested in the lifelong effects of these exposures and these conditions. Dr. Ojo is going to talk to you about post-traumatic stress disorder today. And one of the things we are very proud of with our PTSD program is that we have a model that demonstrates changes in the mouse behavior and changes in the mouse brain months after the traumatic exposure. And we believe that that's really critical in terms of that model being relevant for our PTSD patient population. Similarly, with our Gulf War illness work that you're going to hear about from Dr. Leila Abdullah, we look at mice that have been living for a year, maybe 2 years after their toxic exposures, because the folks we're trying to treat were exposed in the Gulf War conflict of 1990, 1991, and here, 26 years on, they're still suffering from those exposures. So we model all of these conditions and more, but these are the ones you're going to hear about today, and this is the goal. Everything we do at the Institute is about identifying better targets for better treatments, or any treatments at all, in fact, and better diagnostics. And I've got some exciting news. I'm actually just the, what is it, the pre-game show? Is that the proper term? I don't know what. So I'm going to be talking just a little bit about Alzheimer's disease before we get into the main program. And I'm talking about Alzheimer's disease because, as I mentioned, our work here began because of our Alzheimer's findings. So our Alzheimer program is furthest along. But the Alzheimer program here serves as the roadmap for everything else we do at the Institute. Everything is focused on an end game in the clinic. Everything is about treatments and better diagnostics. And we've got that far along with the Alzheimer program. So sadly, I think everybody is pretty familiar with Alzheimer's disease these days. Most people have either been directly exposed to it in their family, or they know somebody else who has Alzheimer's disease. It's a devastating neurological condition, a deterioration that is really devastating in families. These are old statistics now. 1 in 8 people around about the last decade, 2000 to 2010. And by the time we get to 2050, 1 in 5 people is the estimate of Alzheimer's disease. And of course, we're all at risk for Alzheimer's. As we all get older, it's a 1 in 20, sorry, not 1 in 20, a 20% rising to 50% risk of Alzheimer's disease as we move from 70 to 80 and older. So Alzheimer's disease, there's 2 main proteins that we know are critical in Alzheimer's disease. One of them is this protein, amyloid. And we've done an awful lot of work on the amyloid protein. It was the gene for the amyloid protein that we identified those early mutations in back in our early work in London. And amyloid is a protein that does something normal in the body. We think it may have a role in blood vessels, but it starts to build up in the brain as we age. And as it starts to build up, it starts to have toxic consequences. And it usually gets cleared out of the brain into the blood and into the cerebrospinal fluid. But as we age, everything starts to wind down a bit. Things don't work as well as they did when we were 20, I'm sorry to say. That's my experience anyway, maybe, you know. So the amyloid starts to build up in the brain, and when it starts to build up, it starts to have these toxic effects on the cells around. And it triggers inflammatory responses. And inflammatory responses are good. They're a defense mechanism that our body has and our brain has against things that shouldn't be there and against problems that are happening. But if those neuroinflammatory responses persist or get out of control, they start to turn against healthy cells. And then we start to get degeneration and deterioration. The other protein that's important in Alzheimer's disease is a protein called tau. This is a neurofibrillary tangle. On the previous slide, these are amyloid plaques. This is the buildup of amyloid around the neurons in the brain. And here, this is actually inside a neuron. Tau is a protein which is part of the normal architecture of the brain. If you think of the neurons as being like the interstate, and we've got traffic going from one end of the interstate to the other, from Naples to Tampa, say. If you start to get problems happening on the interstate, which of course never happens, but let's just say around University Parkway, we've got a build up there, then things that should be getting from Tampa to Naples don't get there in time, or maybe they don't get there at all. If something blocks off all of the lanes, things can't get through, so the messages don't get transmitted. That's what's happening in Alzheimer's disease. We start to get this deterioration and dieback of the neurons, so the messages aren't getting through. So what we found many, many years ago were mutations in the amyloid gene that cause Alzheimer's disease, and they result in an increase in the production of amyloid. So you can get Alzheimer's and get a buildup of amyloid as you age because our normal physiological functions are winding down. But if you're genetically set up to actually make more amyloid to begin with, then it starts to build up earlier. So these were folks who were getting Alzheimer's disease in their 40s and 50s, and you could see a very clear genetic transmission through those families. Very rare, but told us how central amyloid was to the Alzheimer's disease process. And what we discovered was that the amyloid deposits were associated with those tangles, the tau protein, forming those tangles, those blockages in the neurons. And by identifying those genetic causes, we were able to create animal models of the disease. So we were able to introduce those changes in the DNA into mice in the laboratory, and the mice are born normally and they develop normally, they grow up, but as they age they start to develop cognitive problems, memory problems. And Dr. Scott Ferguson will show you, as you go around the labs today, he'll show you how we test mice for their memory. Everybody always says, how do you know if a mouse is losing its memory? We have tests so we can do that. They also develop amyloid deposition in the brain, amyloid plaques, just like human beings do. And we also see changes in tau protein and in neuroinflammation, excessive neuroinflammation in the brains of these mice. So then the story changed with the Alzheimer field. Now that we had these mouse models, now the game is, can we cure the mice? Because if we find something that can cure the mice or treat the mice, it may be translatable to the human patient population. So I'm only meant to be talking for 10 minutes today, so I'm going to jump forward 15 years at this point and not give you all of the nitty-gritty that goes before, but suffice to say we have looked at thousands of compounds to try and find a treatment, try and find something that would improve the mice, that would block the amyloid production, help clear it out of the brain, address the other issues that are going on in the brain. There's lots of other things go on in the brain of an Alzheimer patient. It's not all about amyloid. But long story short, we have a drug called Nilvadipine that lots of our work showed might be useful in Alzheimer's disease. It's actually a drug that has been available in Europe and Japan for about 20 years or more as an antihypertensive treatment. But our work showed that in those mouse models, we were able to block the cognitive decline in mice treated with Nilvadipine. And we were also able to block the amyloid buildup in the brain. And we also showed that the amyloid actually came out of the brain more. We showed reduced neuroinflammation, and we showed reduced tau phosphorylation. So all of these different things that looked as if this could be a good treatment for Alzheimer's disease. So we conducted an early study over in Ireland where the drug is available on prescription, and I have 1 or 2 contacts back there. And we did a trial just to see about safety and efficacy. Safety primarily, we wanted to know, is it okay to give this antihypertensive drug to Alzheimer patients? Because of course, as Alzheimer's disease progresses, folks tend to develop hypotension. So we have to be careful giving an antihypertensive drug to somebody with low blood pressure. But the truth of the matter is this is actually not a very powerful antihypertensive drug. We think this is one of the reasons it was never introduced into the United States. It could never have competed with amlodipine, which is sold as Norvasc. It wouldn't have been able to compete. So our trial showed safety, showed it was fine to give this to Alzheimer patients. There was actually another completely independent study from ours that looked at folks in Japan with hypertension who were being treated with either Nilvadipine or Norvasc. And very interestingly, the folks with amlodipine all progressed from mild cognitive impairment to Alzheimer's disease, but many, many fewer of the individuals who were taking Nilvadipine progressed to Alzheimer's disease during the time of the study. So that was just independent validation that we might be on the right track. So this is just one, I'm just showing one picture from the mice here. This is the Alzheimer mice that were treated with placebo. And all of these gray patches you can see up here, this is amyloid plaque in the brains of those mice. So these are mice that are actually 20 months old, and they started being treated with Nilvadipine when they were 10 months old. This is considered an active treatment regime. At 10 months old, the mice already had cognitive problems, and they already had some amyloid in their brains. The placebo group still have a lot of amyloid. It's carried on building up. But look at what's happened with the Nilvadipine group. You can see, I hope, even from the back, that there's much, much less amyloid in the brains of these mice. And these mice showed improved cognitive performance. Everything else looked good with the amyloid treatment in these mice. So, on to our clinical trial. Our clinical trial began in Europe in 2013, and it's funded by the European Union. We were able to do the trial there because, as I mentioned, the drug is available in Europe, so we were able to move forward quicker there. And this is a double-blind, placebo-controlled, randomized trial, just run exactly the same way as if it had been done here in the United States. 510 Alzheimer's disease patients. It was run across 23 clinical sites in 9 countries. And so we've basically got 250, 255 individuals who took either Nilvadipine or were on placebo for 18 months. And the trial was fully recruited in April of 2015. The last patient completed in Dublin around about this time last year. And when that finished, then there was the data cleanup and the locking of the database and the analyses. And the results are coming out at the end of 2017. Well, the end of 2017 is now. And I'm going to leave it there. Because after you'll hear the talks on traumatic brain injury and post-traumatic stress disorder and Gulf War illness and our clinic, I've persuaded Dr. Mullen to come and give you an update on our study. Now, I will warn you, we're not really meant to be talking about it yet. So we're going to have to make you all promise not to tell anybody, or else we're going to have to keep you in the building and we're going to have to train you how to work with the mice. Either way, I'm good. You know, we're always looking for good hands. So I'm going to leave it there and move on to my colleague, Dr. Benoit Mouzon. And I just want to thank you again for coming. Welcome. We honor our veterans and our active duty soldiers. Thank you so much for coming, and I hope you have a great day.
All right, so the topic of today, my topic is traumatic brain injury. So this is what we call TBI. So if you hear in the news TBI, concussion, that's all in the same umbrella, under the same umbrella. More precisely, we will talk about concussion. So there's different type of TBI. This is the definition of TBI, an external force arising from blunt, penetrating, or acceleration, deceleration. So if you are in the military, you can have blast injury, an IED on the side of the road and you get blasted. And then you can also have penetrating brain injury, which is, let's say, a bullet who hit your brain. And you can also have concussion, which is more like if you are in a Humvee, a blast injury, and then your head accelerate and decelerate. So it makes your brain move inside your skull. And there is stress applied to the brain, and it creates some different type of injury that I'm going to show you later. So I want to start with that slide. And here you have a child who has a brain injury. This is one type of brain injury. An Eiffel Tower basically fell on a toy Eiffel Tower. So the point I'm trying to make here is there is some type of brain injury who are visible, and you seek medical attention. If your grandchild or child is running around with a little Eiffel Tower on his head, you will go to the hospital, to the ER, and you will seek medical attention. Now let's say the same child fell on a step. You have a 2-story house, he fell on a step, he get a concussion, but you don't see any type of, there is no Eiffel Tower, there is no bleeding, there is no major sign of trauma. However, inside his brain, you might have the structure of the brain is injured, and this we don't see. And it's because we don't seek medical attention, we don't go to the hospital, and the child could be injured, and this will last until the end of his life. So in that situation, they took the child to the hospital, he got released within 48 hours. They removed the Eiffel Tower, antibiotic, fine, no long-term consequences. In the other case, if you have a child who has a concussion, he might have significant problem for his whole life. Most of the time they would recover, but sometimes we don't know why. Some people are more affected by others after a concussion. And this is what you see also in the NFL. You see the football players, some of them are fine and some of them they have neurological disorder. You don't know why they are angry, they kill themselves. Why some are more affected than others. So this depends of the individual, the genetic background, etc., etc. So we will talk about concussion today because most of the type of injury in the military, 85% is concussion. So that's the mild form of TBI. And this is a big problem for the VA and for the DoD. You can see, so it's different type of combat now. It's not like World War II or Vietnam War. So anything, the Gulf War and Operation Iraqi Freedom, anything related to Afghanistan and Iraq, you have a lot of concussion type of injury. So, and this is mixing with PTSD, so you can have a concussion, you have a trauma at the same time because you have the explosion, you see your comrade who are injured, so you have a mix between PTSD, TBI, and then also Gulf War illness and TBI who can be interacting together. So it's very complicated. So the concussion is an issue, and don't wait for the autopsy, because right now, the NFL, what I want to bring with that slide is, the NFL did a good job hiding the concussion problem, because now everybody is aware. They got the public attention, and this brought attention also more research to be done in concussion, and this is also helping the veterans, because without the NFL, probably we will not care about the concussion in the military population. So the NFL did a good job to hide it. That is good for you because now there is more research. So what Barack Obama, our last president, he did a brain initiative and he allocated about 65 million to study the long-term effect of mild traumatic brain injury or concussion. This was done a couple years before. So what I want to show you here is we are part of that consortium. It's called the Chronic Effect Neurotrauma Consortium, CENC. So I can give you the website at the end of the talk or later on. You can go in, there is different studies. You can enroll, there is an imaging study, there is a biomarker study, you can give your blood, they collect statistics out of you. So it's more than 20 different VA across the US. And everything is on the website, you can look it up, they publish, you can know what's going on in terms of research. So Tampa VA is involved in one of those studies. So now I'm going to show you what's going on if you get a concussion, because nobody knows really how does it work, what's happening to your brain if you get that type of injury. So that's a pig, this is a pig brain, and that's a pig model of rotational injury. They do that at UPenn in Pennsylvania. As you can see, pre-acceleration, when you accelerate the brain, the whole structure shifts inside. So you have the gray matter, which is the darker part of the brain, and the white matter, the white part. It's a different type of structure. Think about the white matter is a little bit like squid, if you go to Thai food, and the gray matter is like the tofu. So different physical properties. Some are more resistant than others, and some are more susceptible to the injury than others. The white matter in the middle is very susceptible to concussion, acceleration, deceleration forces. And I'm gonna show you what's happening in the brain. So that's a small video, and that's a neuron, and I'm gonna push play. Let's see. And let's say, so we are not on the field in wartime, but let's say you go back home and you have a car accident. I hope not, but. And what's happening, if you have acceleration, deceleration, what Fiona talked about, those highway who connect Naples to Tampa, that's like a highway. It connects neuron to other neuron. You have cargo molecule information to move along. So now accelerate, decelerate. And you have those structure, those roads who are broken because of the forces. And when you break those structure, those little block, the road block, they aggregate together and they create tangle or amyloid plaque. And if you kill all of those roads after concussion, you damage all the connection around the neuron, and there is no more road, so the neuron will die because it doesn't get any more information or no more food. Do that over time. Let's say a football player, few seasons, they keep having concussion after concussion. Or if you are in the military, you do basic training, you do boxing, there is a lot of concussion during the training. Well, at the end, your brain, you keep killing those connections, and at the end, your brain, you have a brain atrophy. And your brain will look like an Alzheimer brain. Except this can happen in your 50s, 60s, instead of happening a little bit later for Alzheimer. So the pathology we see with TBI is very similar to Alzheimer. However, the origin is different. This can be prevented because it's concussion. So you don't, if your child play football, he get a concussion, you don't want to let him play again. You want to wait. I'm going to show you why in a minute. So what you have seen in the movie, it's called diffuse axonal injury. It's a type of injury. On the top, you can see this is a control subject. It's an 18-year-old female who died from cancer. So everything is normal. You don't see any, the brain staining is what is abnormal. The brain staining is an axon. If you look at here, this is an assault. So this is a brain from Scotland, so bar fight. You see, and somebody died resulting of a blunt trauma to the head. And you can look at that structure, it looks like a spaghetti, a brown spaghetti. That's an axon who has been injured. And you have those proteins who start to accumulate, and we can detect them. That's why we see the brown staining. And if you look at 22 hours post-injury, that's a car accident, another subject who died. And you can see those axons that start to do a beading process. You see how it makes like a little ball? And it's because when it starts to swell, and then it breaks because it's so swollen, it just cut. And this is how you lose your connection between each neuron. Again, that's the same thing you have seen in the movie on the previous slide, but this is in a Petri dish. We can grow axon. And what we do some kind of, to do a concussion, we do a blast wave, a little blast wave, it will stretch the axon. So it's like a blow on the axon. And you can see the beading process. That's normal shape, you see, it's a regular rod, but you can see the swelling here and here. And this is when the rods are broken inside. You see that black line? If it's broken, so everything keeps coming in, and then it starts to swell. If it swells so much, it breaks. Again, that's why we say, if you are basic training, you get a bad concussion, while boxing or practicing, or your child, grandchild, they get a concussion on the field, you don't want them to stay on the field, you want to take them out. Because it's mild injury, so some of them are intact. You see this one? It's still alive, it's not broken. So if you give some rest, it will pick up the swelling. If you give a couple of days, a couple hours, days, it will pick up the swelling, and maybe you will not lose your connection. So that's why we're trying to push now in the news you have seen in sports. We don't want the player to stay on the field. We want to take them out. So if you have a concussion in your family, just don't do any physical activity anymore. You can do physical activity, but that doesn't result in a brain trauma. So go to the gym and do the bicycle, that's fine. You can play video game, usually kids prefer. So you can do that, that's fine as well. And you need to rest. You need to give time for your brain to heal. And just to give you an idea, because TBI is also, that's the brain network, how your neurons are connected together. That's a control subject. You can see it's very thick, it's very bushy, a lot of connection, a lot of rods. This is an injured patient. A lot of connection who are dead, missing. So if you look at TBI, and that's why if you think about the football player and some of the soldiers, their issue, mental issue, it's not like they don't remember, but they will have difficulty to concentrate. They have difficulty to read. They are mood disorder because they can't sleep. Because of all, the network is damaged, so it creates a burden for the brain to process all the information, and they get more angry and sometimes they burst. That's part of the reason why the people are like this. So we have to, there's still a lot to learn out of this. And in terms of brain imaging, it's very expensive, and you need a very good scanner to look at that, and it takes a lot of time. So it's not that everybody is doing that type of brain imaging. Anyway, so what we do here at the Institute, we mimic concussion, because as you know, if you get a concussion, you don't die unless you have a polytrauma, something cuts your throat or something else. So we don't have brain tissue to look at, not a lot, maybe 200, 300 total across the world. But we compare, so with the mice, what we can do, that's the way we do that's an injury apparatus, so how we do the brain injury in the mice, it's an impactor. We impact the head of the mice, and then it compresses the brain, and then we euthanize the animal and we look at their brain. So what we did at the Institute for the last 3 or 4 years, we do single injury or multiple injury when they are young, and then we sacrifice the animal at 24 hours or 6, 12, 18, and 24 months. So this way we can do timeline. We can look at what's going on in the brain at all of those different time points. So we will collect the blood. We can collect the brain for pathology. We also do lipid analysis out of the brain or the blood. And we also test the behavior of the animal. Do we have issue in memory issue? Do we have executive function? Learning issue? They have a lot of learning issue, actually. They learn slower, the mice who are injured. It's not they do not remember, but they have anxiety issue, and they have also learning issues. So it takes more time for them to learn a task. That's it. And based on what we see in their brain or the lipid profile, we can look back and say, we want to treat that problem we see in the brain, we want to remove it. So we will look for drugs who can alleviate that problem. So let me show you. If you look at that picture here, that's a mouse brain, and if you look at the brown staining, that's abnormal. So that's a mouse. On the left here, you have a control mice. This is it's a 24-month animal. And this one received 5 injuries at 3 months of age, and then we let her leave the mouse for 24 months. And we can see this is abnormal. You can see that it's called inflammation in the brain. So you want to remove that. So between the 3 months of age, then nothing happened to the mice. We let her leave, she can do whatever she eats, do nothing, no more injury, and you can still see, for every mice I injure, I will see the difference 24 months later. The one who received 5 injury, 5 mild injury from the control. And this is what it looks like if you do a timeline, the inflammation in your brain. You receive injury, so it kind of shifts everything, and then you have always more inflammation in your brain compared to the control animal. This is the blue line, this is the control. The black line is single injury, and then the red line is multiple injury. The issue we have is concussion. It's so mild, we don't realize that our brain is injured, because it's not like you lose your arm and it's missing, you see it. So concussion is a lot of small problem who adds up through your life. But it's like the vision problem you might have. You don't realize that, but the veterans we see, they have a loss of visual field due to the injury. But it progresses over time very slowly, so you don't see, it's not right away. So that's an issue. If you haven't seen a mouse brain, this is what it looks like under the microscope. So it's a sagittal section. You have the brainstem, the cerebellum. This is the hippocampus, the cortex, and then you have the corpus callosum, which is an area which is very sensitive to brain injury. It connects both hemispheres, and there's a lot of road in that region. This is the human brain. So here you have the corpus, the cortex, you have the gyre, the cerebellum, brainstem, different structure, but it's comparable. So what we see, if we compare mice to human, because everything we do in a house has to match what we see in human, or it's a waste of money, there is no point to study. So we have always to compare what we see in mice, do we see it in human as well? And it does match for most of it. So inflammation, that's a controlled human brain. You can see there is more brainsteming here. This is 4 years post-TBI, 40 years, you see. Axonal injury was what I was talking about, the axon who swell. We see that in human, same time frame as well. Those little bleeding process, those are the swelling. And we also have, because you have axonal injury, you have demyelination. It's like multiple sclerosis. The myelin disappears. We see that too in a brain-injured patient, in the white matter. So that's a control subject, and you can see it's very obvious, the loss of myelin in the white matter. So this brings us to us. So you are here because you can help us to, when we talk to you, we know what type of injury you have. It helps us to get us idea where we can focus our research. And there is different, this was a study was been completed so far, but we can collect the blood from you. You can give feedback to us. So let's say for that study, we collected blood from soldiers before deployment, during deployment, and post-deployment. And then we can look at biomarker. So when they come back, we can compare the one who received TBI versus the one who don't have TBI. We can compare the one who have PTSD versus the one who don't have PTSD. So now if you want to, Dr. Keegan will talk about that later, but we are recruiting for PTSD, Gulf War illness, and I think TBI. So if you're willing to give your blood, we can look at those little molecules in it and see if we can compare to see if there is an injury effect versus a control effect. And maybe we can do testing. Later on, it will help us to say, you got an injury, at the hospital we collect it, and we look for that marker, and it tells us, yes, you got an injury, so you're sick. And then you will get the treatment. Unfortunately, there is no treatment for TBI, so it's going to have to wait. On that said, I would like to thank you all, the troops, for your service, and also the family. I have to say that we should be very supportive for the veterans, so you can thank your wife. Because what we see in TBI, it's a lot, people who are injured, if they have family support, it helps them to recover a lot. If they get dropped, if the work environment doesn't support them, they end up on the street. And usually they lose their job, then they lose their wife, or vice versa, and they end up quickly on the street. So the support, the work support, and the family support is very important for people who have PTSD and TBI as well. All right, on that, I think I'm done, and I will hand the microphone to Dr. Rojo.
Thank you very much for coming to the VA open house at the Roskamp Institute. My name is Joseph Ojo, and I'm going to be talking to you over the next 15 minutes on post-traumatic stress disorder. So when you look at different types of trauma, there are certain types of trauma that show very high prevalence rates of PTSD. And I've highlighted the last three here. You can see that combat exposure actually has one of the highest rates of prevalence of PTSD with almost 30%. And childhood abuse as well ranks high with 45%. And rape over 45%, between 45 and 50%. Another important aspect, which is worth mentioning, is the quality of life in PTSD. There are different issues that can compound on the quality of life of a patient with PTSD, either from premorbid issues as well as concurrent and long-term issues. So some of the premorbid issues here are poor quality of life, lack of social support, poor psychosocial resource base to support the individual, and material resources as well. With respect to the concurrent issues, there are significant influences that can compound on the quality of life of a PTSD patient as a result of things like alcohol and drug use, sleep disorders, physical illness, and other comorbidities. And there are long-term issues as well, such as employment, job skills, and damaged social ties and things such as that. So any focus on the war zone trauma, there was a study done recently looking at the US and UK military records, and they were able to collate the data together, and they found out that there was around 19% of veterans who developed war-related PTSD during their lifetime as a result of exposure to combat. And from those that they continued to study from the time of original assessment, they found out that 9% of them continue to have PTSD. If we rewind the clock back to the Vietnam War, there was a study done 10 years after the Vietnam War. They observed 28% of those involved in combat were diagnosed with PTSD, and a follow-up after 40 years showed 11% were still experiencing PTSD symptoms that were significantly impacting the function of the patient. So if we go back to the recent conflicts in Iraq and Afghanistan, there's even a large numbers, 300,000 of those involved in that conflict have been diagnosed with PTSD, reportedly diagnosed with PTSD. Military suicides as a result of that conflict has been shown to be at the highest rates in the last 10 years. And national suicides as well have been completed by veterans as a result of the increase we've seen in PTSD diagnosis from those involved in that war. And interestingly, in 2012, both in the US and in the UK, there were more deaths as a result of suicides than in combat. So this pretty much highlights how serious an issue this is. So what are the challenges that researchers and clinicians have to overcome with respect to work done on PTSD? The first is the proper diagnosis of PTSD. It can be quite clinically complicated and complex because it can happen with other mental illnesses, and especially with PTSD and traumatic brain injury, like Dr. Benoit Musin just talked about. Typically you see overlapping of symptoms between people exposed to traumatic brain injury and those exposed to trauma, a stressful event. And there's a great push by researchers to identify biomarkers as well as non-invasive biomarkers that can help to diagnose the patients quicker, more accurately, before the negative symptoms emerge. I'll put this slide here because it links a little bit with what Ben talked about, and this is to do with CTE. So I'm sure you've heard about all the recent media awareness of CTE from, no? Okay, CTE is chronic traumatic encephalopathy, and it's a neurodegenerative disease that happens as a result of repeated blows to the head, concussions to the head. And what happens is you can see, this is a control brain, a normal brain, and you can see a person with CTE in advanced stages. And what happens is that in this groove regions in the brain called the depths of the sulci, you find accumulation of this proteinaceous, this protein called tau, aggregated into clumps in the depths of the sulci. And if you look closely with a higher micrograph image, you find this tau proteins around the blood vessels. And this can be found in different brain regions that can significantly compound on certain functions. And typically the clinical profile of people with PTSD, it ranges quite a lot. So you can see different types of symptoms, from alteration in behavior and mood, also motor dysfunction, and also you find cognitive deficits as well. And here you can see some of the different symptoms: depression, suicidal thinking, paranoia, anxiety, quick temper, confusion, and so on, impaired judgment. So it was Dr. Omalu who brought this condition to light with respect to the NFL, but it's been known for many years before that, almost 80 or 100 years before that. So recently, over 6 years ago, he reported one of the first cases of CTE in an Iraq war veteran who suffered from PTSD, and he committed suicide. And if you look at this micrographs here, you can see this is actually taken from the brain of this individual. You can see the deposits of these tau proteins forming around these brain regions right here and significantly affecting certain functions. The other challenge is to find efficacious therapeutic approaches that can help to treat patients better than what we have right now. PTSD, especially combat-related PTSD, can be quite refractory to current treatment approaches. So that's one thing that needs to be done. It's a huge burden on healthcare costs, and we need new perspective on the biological mechanisms that are involved in PTSD. So this is a simplified diagram here showing the neurobiological basis of PTSD. So you can look at the patient right here. He's exposed to a stressor, and this could be someone who has just witnessed something on the line of duty, or it could be someone exposed to rape or whatever the case. But there are certain regions in the brain located here, the cingulate cortex, the hippocampus, and the amygdala, that immediately react to this stressful stimuli. And they are involved in processing this information and consolidating this traumatic fear, as well as the things that are associated with the environment. So what happens is that these regions are activated to store and process this information. And there's a region in the brain called the hypothalamus that sends signals to another region at the base of the brain called the pituitary gland, which releases hormones into your bloodstream. And once these hormones are in your bloodstream, they activate an organ right on top of your kidneys called the adrenal gland. And the adrenal gland releases more neurochemicals, adrenaline and cortisol. And both these neurochemicals can activate the systems throughout the body, from the cardiovascular system that regulates your blood pressure, your heart rate, blood flow to the brain, as well as the facilitation of energy from glucose metabolism to immune system issues and so on. So what happens in a normal person is that this circuitry returns back to normal. But in a person with PTSD, there is a disconnect somewhere, and there is a dysfunction, and this is what we're trying to try to understand, get to the heart of what underlies this dysfunction. So what are the studies we're doing here at the Roskamp Institute? We've conducted recently a study looking at active duty soldiers, following them on a prospective type design study where you look at different time courses, so pre and post deployment. And we've collected blood samples from these patients, these soldiers, and we've interrogated them with our techniques to look at changes in proteins, lipids, and also the genetic background of the patient, to try to understand what are the molecular correlates and indicators of some of the changes we see. Why do some active duty soldiers return back home normal, and some return back home with symptoms of PTSD? We also have existing collaborations with those who conduct neuroimaging studies, where we can correlate what we see in the blood with what we see in the brain. And also recently, we've been granted access to some autopsy brain samples of patients who died from PTSD. And we're going to be looking at this region right here called the medial prefrontal cortex, which is very important in what I spoke to you previously, to do with the alteration and fluctuation between the hypoarousal and hyperarousal state. So the undermodulation of emotion and overmodulation of emotions seen in PTSD. So with respect to the preclinical studies we do here, we have ways of exposing animals to stresses that can induce a PTSD-like phenotype. And animal models are important, like Dr. Musen just mentioned, because we can manipulate the genetic background to get to the heart of fine details of how certain genes or proteins or pathways contribute to the pathophysiology of the condition. And there are several ways we can expose animals to PTSD, like phenotype. We can expose them to physical trauma in the form of inescapable foot shocks, predator exposure, psychosocial stresses, as well as traumatic brain injury. There are also ways that we can monitor the behavior of animals to look for indices of anxiety, for example. In this test called the elevated plus mace test, where you place the animal in the middle of this maze with a closed arm and an open arm, and the time the animal spends between the closed and the open arm is measured. An animal that shows increased anxiety would tend to spend more time in the closed arm as opposed to the open arm. We can also use a test called the forced swim test to look for signs of depression, where we place the animal in this jar, 3/4 full of water, and they won't sink ever, but we would measure the time they spend immobile. And an animal that shows depressive-like phenotype would tend to give up straight away once you drop it into the water. We also have tests to look for indication of social behavioral deficits in this 3-chamber test that monitors the behavior of animals. And also we can monitor sleep cycle and a host of different other behavioral parameters, which I haven't presented here, and I'm sure Dr. Ferguson will be talking about, who you're going to meet with the lab tour. And we have a repertoire of different techniques ranging from histopathology to proteomics and lipidomics, as well as epigenetics, looking for changes in DNA, proteins, and lipids. And we can interrogate different brain regions, as well as blood samples, as well as peripheral organs like the adrenal glands, to try to get to the heart of what is the biological responses, what underlies these biological responses in PTSD. And how will our research benefit the military population? We're hoping that we can better understand the pathophysiology of PTSD. We're hoping that some of our work will be able to identify early fluid biomarkers that correlate with PTSD, that can distinguish resiliency as opposed to susceptibility and predisposition to PTSD in certain active duty soldiers who return from combat. We're hoping that our work will facilitate early detection of patients who are at risk of developing PTSD before the negative symptoms occur, and also identify new therapeutic targets for drug discovery. So what can you do to help us with our research? You can visit our clinic and participate in whatever clinical studies that are going on, and you can give us details of your past history as a veteran. We're interested in knowing, and also donate blood samples for clinical research. And with that, I'd like to thank you all for your honorable service and hand over to Dr. Abdullah.
I'm going to take you back to 1991. I'm going to talk about the Gulf War illness, and I was lucky to get my PhD under Dr. Fiona Crawford and Ganya Eid Ghazala. I did my PhD on Gulf War illness, and I stayed at the Institute because I wanted to keep working, and I wanted to use that roadmap to find a treatment and figure out how we can treat this illness. So some of you may have already been in the clinic and participated in the studies. So I just wanted to briefly mention Gulf War illness. It's when soldiers returned from the 1991 Gulf War, no one believed them. They started reporting symptoms that didn't fit into any particular category, and people started saying, well, this isn't real, this isn't real. And as a consequence, it's taken us so long to recognize that this illness, it's a multi-symptom illness, doesn't fit any of the criteria of the regular diagnosis. but it's a cause of great distress to veterans who return from this Gulf War and are suffering from this illness. So I'm dedicated to making sure that we can help diagnose and help treat this illness. So you've heard our roadmap at the Institute here, that we're focused on finding treatment and we're focused on developing objective biomarkers. And that's my focus here too. As part of my PhD studies, I used the chemicals that Gulf War veterans were exposed to develop a mouse model of this illness. And the reason it's important is because we wanted to make sure that we can use this model to test therapies that then we can then take forward into human studies. We wanted to make sure if it's going to fail here, if it's not going to work in the mice, it's not going to work in the humans. So this is our first step to making sure that we only take forward treatments that are going to benefit the veterans. And right now, there isn't an objective blood test available. So there's a great effort. There's GWI Consortium. They help collect blood samples. We're collecting blood samples here at the clinic. We want to look at blood changes, develop a blood test that then we can use for diagnosing this illness and help make sure that the veterans get the care and treatment that they need. So my initial work focused on developing the Gulf War illness mouse model. I used pyridostigmine bromide that was used as an anti-nerve agent to protect against nerve gas exposure, and pesticides. There's a combination of pesticides. I focused on permethrin that was embedded in the military uniforms. It was applied. It was very freely used. So I used a combination of these two to develop a mouse model. We did a very short exposure, 10 days exposure to these chemicals, and then we looked at long time after the exposure, 5 months, 16 months, 11 months. What consistently popped out was, if you look at this is, again, points to inflammation. These are glia cells, astroglia cells they're called. What they do is they provide support to the neurons. They take the nutrients and the supply that comes through the blood into the brain. And you can see here that they're highly dysfunctional compared to a healthy mouse's brain. And that's happening because they're not doing their job. They're not supplying the nutrient. They also take part in reducing inflammation. They're not doing that. And as a consequence, when we do the behavior testing, which is, you've heard Dr. Ojo and Dr. Moazo mention, this test, this is a memory test that we do. We train the mice to find a box that they can escape into. So you can see the blue line are the mice. This is 16 months after they've had the Gulf War exposure. And you can see that the blue line is the healthy mice that didn't get the Gulf War chemicals. They seem to quickly learn, the line being shorter here. But the red one, these were the mice that had been exposed. You can see that they're not learning very well. In fact, they just sort of plateau out over the days. And when we test their memory, long-term memory, so we train them, then we take the hints away, and then we'd let them search the platform to figure out where the escape route is. And you can see that it's taking them much longer. The red bars are bigger, so it's taking them much longer to find compared to the healthy mice. So even after such a short exposure, this is 16 months out, 22 months out, no matter what time point that we've looked at, the Gulf War chemical exposed animals continue to perform poorly on tests that relates to memory. So as I was doing this work, I started to focus on molecules called lipids. What they are is fat. A lot of it comes from your diet. You must have all heard of omega-3, omega-6 lipids. So a lot of these come from your diet, but your brain needs it. So just to give you, this cheese here practically represents your brain. There's so much fat in it, and it's because it's needed for cushioning, it's needed for support, it's needed for giving it structure. And so I thought it was a very important area, and I wanted to understand how that applies to Gulf War illness, so I started focusing on that. So just some more information about the lipids or fats. The saturated, these are very rigid fats, and they come from cheese. It's mostly in there. Good fat, the omega-3, comes from salmon, sardines, very healthy fish, oily fish. And then the unsaturated, the omega-6, it can be good or bad, but mostly if it's too much of it, it's bad for you. And then that comes from your red meat. So just in terms of what you eat gets into your brain, and these are some of the lipids that get into your brain. So thanks to the GWI Consortium, Dr. Klimas and Dr. Kimberly Sullivan, they're involved in that. They sent some blood samples a while back to me. And we've been looking at these blood samples from Gulf War veterans who have the disease and who don't. And what we wanted to understand is that what are the changes in the blood that we could potentially use for diagnosis. We can develop a blood test that can give us an objective indication of what the changes are. So the omega-6 and the omega-3 theme consistently seems to crop up. And so we've published before where we looked at mouse models of GWI that we developed and from other investigators, and then samples that we were provided from the GWI Consortium. And what we were seeing, and I'll just point it to this 22.6, what is called. This is the DHA. So you all know what DHA is? DHA is the omega-6 fatty acid lipid that comes from fish. It's very long, and it's the primary nutrient that's in the fish, but only the fish that is in the sea, not the farm raised. If they don't feed them the right quality of material, then they don't make DHA. So what we can see is that, so what we're looking at here is the free form of it. You want the DHA to be in bound form in your intact lipids, but here in the free form, it's not as useful as it is in the bound form. So you can see that in the red bars are blood samples from veterans with GWI, the blue bars are from Gulf War veterans who didn't have GWI. You can see that these levels are much higher in the GWI. So that means that it's not being metabolized properly, maybe, it's not being utilized, it's not where it's meant to be. And same thing with number of others. You can see that there are a number of other these lipids that are increased, as well as the very long chain. So the relevance of the very long chain lipids is that they are again needed for the brain for the support of the neurons. But what happens is that if they don't get in where they need to be, they accumulate. And that indicates that there is a disconnect in how they're broken down in the body. So this points to us that there is a problem, metabolic problem in GWI, that these lipids are not being broken down how they should be and utilized. So this is one of the areas I'm trying to increase the sample size. We can replicate this, validate this. So if any of you have not participated yet, who've been in the 1991 Gulf War, please sign up. We have a Sandra sitting back there waiting for you to sign up. So, and thanks to the consortium, we're still getting some samples to increase this. So we'll jump over to, so how can we use this information? What can we do to figure out, how to correct that problem? So treat lipid with lipid. Simple, right? So we are looking at figuring out how to correct the lipid imbalances, how to make the body to make sure that it breaks it down, and then what happens? Can it then correct all the problems that the veterans may be suffering from? So I'm focusing on this little lipid called oleal ethanolamide, and for short, OE. What it is, it's it's derived from oleic acid, olive oil, and it's found in numerous dietary supplements called, it's in cocoa powder, oatmeal, millet, but it's probably not in enough quantity. So, so what we're looking at is, figuring out, okay, how can we make it? How can we produce it? We're looking into that. We have some more work to do. But I wanted to test that here in our GWI mouse model to see, hey, is this going to work in our mouse model? As I said earlier, if it doesn't work in the mouse model, we're not going to take it forward. So we developed our mouse model, 10 days exposure, as I described earlier, very short with the Gulf 4 chemicals. And then we administered OEA in their food. We just mixed it up in their food and gave it to mice for over 6 months. And throughout that period, we tested the mice to see how their memory was. And we looked at their fatigue-related parameters, which was, it's a forced swim test. So basically, mouse is allowed to swim for 6 minutes. And if it starts to float instead of swimming, then we know that it's getting fatigued and is showing some of the exhibit. And fatigue is one of the major issues that affect the veterans with Gulf War illness. Obviously, this is in my study, so we don't know for sure, but that gives us somewhat of an indication. So, do you remember going back to the Barnes maze test, teaching the mouse to escape into the hole? So again, these mice were trained to go into the hole and escape, and so they were learning in the 4 days. And the red line here shows a mouse that was exposed to Gulf War chemicals. The orange line here is the mouse that was given OEA in a dish after it was exposed to Gulf War chemicals. You can see that we've got a super mouse here. It is learning so fast, it's doing better than even the healthy controls on finding and escaping into the hole. So it's learning very quickly. And you can see here, after 2 months, mouse that didn't have OEA but had Gulf War chemicals, it's less frequently going to the escape hole, where the mouse that received OEA but was exposed to Gulf War chemicals, it is doing just as well as the healthy mice. So we really think that potentially this lipid, the dietary lipid, may potentially help at least alleviate the memory. And then we did the fatigue test that I mentioned. So you can see that this Gulf War chemical exposed mouse, it's floating. So the immobile time means that it's floating, it's instead of swimming. So you can see it's using that as a strategy to kill time. But here, the mouse that received the treatment is doing well. It's doing as well as the control, the healthy mouse. So again, we think, potentially this is taking care of some of the behavioral problem and memory problem in the mouse that were exposed to Gulf War chemicals. So after that, we did the biochemistry study. So remember those glia cells I was talking about, estroglia, that support the neurons. So these red cells, that red stain that you see here, it's looking for its marker for those glia cells. So you can see that mouse that were exposed to Gulf War chemical, they have so much more staining, like you saw in the earlier slide. So this is looking at the time point here is 11 months after the Gulf War chemical and 6 months after the treatment. So these mice are quite old at this point. But as a consequence of the treatment, you can see here that that's gone. It's very few of the glia cells that look like that they're active. Most of them have quieted down. And you can see this is the cortical region. This is a different part of the brain region. And this part of the brain region is involved in memory processing. So you can see there's a lot more of the red stain here. But after the treatment, there isn't that much of the red cells. So we think that it's potentially reducing the the activation state, the inflamed state of those cells. So what does it mean in terms of the inflammation? So you've heard us talk about inflammation, that inflammation is bad for the brain. So I won't get into the details. We looked at a couple molecules that are involved in promoting inflammation in the brain. So one is called IL-1 beta, the other one's called IFN gamma. The bottom line is these are molecules that promote inflammation in the brain. You can see that it's so much higher in the brains of mice that were exposed to Gulf War chemicals. And after the treatment, it goes down to the same level as, pretty much the same level as the healthy mice. So we think this is potentially worth working on and moving forward. We have a lot more work to do, but I think this is giving us a hint. We are also, as part of our work, we're looking at dietary approaches. We have other programs. Dr. Aid Gazalia has programs for targeting mitochondria, which is the powerhouse. The one aspect of OEA that I didn't get into is that it also corrects the communication, metabolic communication between the powerhouses of the cells that break down lipids. So we're working on figuring out, how this is going to help correct a lot of the features that are prominent in our mouse model that potentially have implications for GWI. So hopefully we have a lot more work to do to move it forward, but we have some indication that this might work. So I just wanted to thank you for being here. Thank you for everything that you've done. If you've already donated blood, thank you. But if you would like to, there's a table there. And and actually, I forgot, this is what I was talking about. Again, you can see that the 22.6 is increased in the brains of the GWI mouse model, the red line here, and it's going back to the normal. So this is what I was talking about, the powerhouse. It's the OEA is making the powerhouse of the cells to work normally now. So thank you for your service.
Our care tends to focus a little bit more on the brain, on kind of things above the neck. So Alzheimer's, mild cognitive impairment, people that have some memory complaints may be related to a traumatic brain injury. You heard a talk earlier about the basic science behind traumatic brain injury. And then Gulf War illness, I'm going to get into just a little more on the clinical side. And then, of course, things like Parkinson's disease, multiple sclerosis, headache. A lot of these things kind of at the basic science level, there's a lot of overlap. So I think some of the discoveries being made here, maybe in a traumatic brain injury, maybe somehow that could also help us understand a disease like multiple sclerosis, which also has inflammation. So there's a lot of overlap between these different diseases. We also do clinical trials. Those tend to be the pharmaceutical companies. Maybe Pfizer has a new medication for migraine headache. They need to pick a number of sites around the country to do those clinical trials. We'll be a site where maybe 5 or 10 people with migraines will come to us. We'll track them after they're either getting a placebo or a drug and see how they improve or not improve. And that'll be kind of proposed to the FDA to see if something can actually be approved as a new medication. We do that in multiple areas, kind of similarly with the treatments we're looking at, the disease states we look at. We also do clinical trials in those areas. So Alzheimer's, MCI. There's also even a push now when it comes to memory. You heard Dr. Mullen talk about kind of slowing the progression. Can we take people in age 60 to 74 who may have normal memory? Can we give them a pill on a daily basis that'll prevent them or slow the progression to dementia? That's kind of the way people are moving forward with this disease, as opposed to, he was describing later on, it's a little bit harder to help in those situations. We do clinical trials on things maybe that help with agitation with more advanced dementia. So there are treatments out there, but they're more symptomatic as opposed to disease modifying. But we have a trial, one of the pharmaceutical companies, Novartis, has a study now that's moving it, looking at a pill to kind of prevent progression to dementia. So this requires a big team. There's kind of two groups. I'm one of the full-time neurologists. We have a number of part-time neurologists, a great nurse practitioner who's really focusing on dementia care, and neuropsychologists and nurse and support staff for all of this. And then the clinical trials, they require people that are kind of visiting with the patients as they come in and collecting information. And so we have 3 of those and a regulatory specialist. So I'm just going to go through Gulf War illness more as a clinical syndrome. You know, you heard more on the basic science where Dr. Abdullah is kind of looking at what's kind of the cause, because there doesn't seem to be a clear definition or one etiology. But these are the symptoms that we hear. This is one of the definitions, the Kansas definition. So there are 6 kind of areas that people complain of, and someone has to have 3 of these 6. So fatigue is a very common component of this, and we have to make sure this person doesn't maybe have sleep apnea causing fatigue, but it's some kind of a more non-specific fatigue description, or pain syndrome, or some neurologic, and then some people have some irritable bowel complaints or gastrointestinal, and all of these have to develop following deployment to the Gulf War. And respiratory, or even some people can develop some nondescript rash that could be associated. So this is the clinical syndrome that we see in people, and that we are now really managing the treatment just looking at the different symptoms. So these are the things we have to exclude, a number of other diseases that can cause it. And we listen to a history. We get a history just like any doctor. We're really hear how someone presents, how people progress. We order some labs to rule out some other causes, like something like an ESR, an inflammatory lab, or an ANA, a lupus type lab. And then we do different evaluations, maybe an MRI of the brain if indicated, and sometimes refer to a specialist. And if all those things kind of rule out other causes, then we have a diagnosis of Gulf War illness. And there's, as Dr. Abdullah was talking about, there's really not one unifying etiology, and that's what people are trying to generate some specific entity to go after, as opposed to just treating the different symptoms. There is a large volume 10 called Gulf War and Health. It's a 275-page book with over 400 references in it, kind of describing and summarizing all the literature in Gulf War, and that's up to date as to 2016. And then here, the Institute of Medicine has a kind of summary of all the treatments, and in there they say there hasn't been necessarily a validated cause. So that's why I think the scientists trying to understand it better is really important. Here's this is kind of the summary of some of the symptoms. This is where, as a clinician, we see all these different medications. Many of them are kind of in the antidepressant range, or cognitive behavioral therapy. So we're looking at the different sub-symptoms. And similarly, the update in 2013 focuses on a list of different medication options, but none of them get after things like what we're talking about, mitochondria, inflammation, the things that the scientists are really seeing that this is actually the cause, and we need to develop medicines that are going after those causes. This slide gets to the complexity, which I think Dr. Mullen was kind of alluding to on a clinical trial, that first there's all this basic science work that has to happen, and then drugs go through phase 1, 2, and 3, and these are in the human, and this is over a 10-year period. The nice thing is that we have some medications now that are being tested. They're already approved, so they've already kind of gone through that phase 3 testing, but are being used off-label. We as clinicians can take a drug, example is Neurontin or gabapentin. It's approved to be used for epilepsy or seizure disorder, but we use it to treat neuropathy. So that's an example. It's already been FDA approved for one indication, but we'll use it for something else. In Gulf War illness, now they're testing a number of medications that are approved, such as rituximab, Enbrel, and then these are more supplement-based, and they're testing that in people to see if it improves symptoms and hopefully benefits them overall. So if people, this is the kind of the referral to Sandra again, or there's a table out front, if people are interested or know people with that were in the Gulf War, they don't have to have Gulf War illness, Gulf War syndrome. They could be actually in there and doing fine. And we also need people with Gulf War illness to come for a part of a day. They give a blood sample, do some memory testing after they consent. That's, we'd love people to sign up. And then also traumatic brain injury. We have a traumatic brain injury study sponsored by a pharmaceutical company that's looking at some agitation that sometimes comes up. And then we have another one that's more on the basic science side, where we measure blood over every year for a couple of years to try to help the scientists go further in what they're studying. So I think that's all I have to say. The example Dr. Mullen gave was an example of taking something in the lab, something that they were working hard at, and then translating it after some time into a human study on an international level and seeing a benefit. And that's what I think this place does a great job of.